Doesn't matter.
It's the water fountain.
So is there a place that they will be able to see me, or are they only going to see the
They
slides?
are seeing the camera back there.
Okay.
Let's see. Yeah, so they can see you kind of walking here.
Okay.
It could, if you like pointing with this,
I think.
or you can use the
I will
mouse.
use mouse.
Yeah.
That will be easier. And then maybe if there's a way to dim it a little bit.
Yeah.
Yeah.
Yeah, I'll give you a little introduction, but then we'll turn those lights off.
So the windows are on.
Yeah.
Thank you so much. I appreciate it.
All right. Thanks everyone for joining online. I know we had a rough start today with the internet down at the Welcome Center, and apparently you can't get in at all if that ever happens. But we got in, and there's enough time to go forward with the seminar. So I'm really happy that our guest drove over today from LSU, Sibel Bargu. She is the Shell Professor of Oceanography and Wetland Studies, if I remember correctly, in the Department of Oceanography and Coastal Sciences. And you also have an administrative role as Associate Dean for your college. So I'm really glad she made time to come see us today. She's an expert in phytoplankton ecology, harmful algal blooms, and she's been doing a lot of work in Louisiana waters. So many of us have a lot of interest in that as well. So please take it away, and she'll be here for a little while afterwards. We're going to go for lunch. But if anybody wants to talk more, we'll try to do it quickly because they have to leave at about 2 p.m. But thanks so much for coming.
Sure, thank you. Is there any way to check if everyone can hear me okay? I guess they can. Okay. All right, thank you so much. It's a pleasure to be here. I was just telling Chris about 15 years ago, and probably many of you were not here back then. And I was talking about Pacific Ocean and neurotoxin domoic acid. But today I will be talking mainly about cyanobacterial problems in Louisiana estuaries. So when you come to a place, you kind of adapt quickly. And Louisiana has no shortage of problems. So cyanobacteria was one of them, so we tackled. And this presentation, I don't know if any of you have listened yesterday's Louisiana Sea Grant webinar. It's a little bit of an expansion from that seminar. So if you did, I apologize. There will be a lot of same slides. But my colleague, John White, and myself, we are both in the Department of Oceanography and Coastal Sciences. We started working in Louisiana estuaries around 2007, 2008. And over the years, we have observed that the changing climate, as well as the implementation of the coastal restoration activities, are changing the water flow and nutrient dynamics. And the combination of these changes are impacting the frequency, intensity, and spatial distribution of cyanobacterial blooms in Louisiana estuaries. So we started kind of focusing on different estuarine systems. But before I start giving examples what we are seeing in our systems, I just want to give you maybe one or two introduction slides. Coastal systems are exposed to a variety of impacts from both natural and human-induced stressors, such as external nitrogen input, changes in hydrological cycles, increase in sea level and water temperature, and the changes in the strength of winds. And in many systems, all these stressors are yielding eutrophication, increasing in primary productivity, as well as changes in the physical environment. And that can result in harmful algal blooms or hypoxia that we know both can cause environmental and public health risks. In Louisiana, the overall productivity of Louisiana estuaries are physically and biologically controlled by nutrient-rich freshwater discharge from the Mississippi River and increasing human population to be able to respond to increase in human population in these land-water interfaces, people made a lot of alterations to this watershed. Levees is one example that people built to protect themselves from flooding. But when they were doing that, they also altered how the river flows. They caused reduction in nutrients and sediments that goes into wetlands, and ultimately caused the loss of wetland areas. So to be able to combat that, and sedimentation and the deposition and the accumulation of sediments are critical to regain these wetlands. And therefore, river diversions become very popular structures in Louisiana to be able to reconnect the river to surrounding marshes and to be able to regain these lost wetlands. But river diversion might have some other ecosystem services such as protection from flooding, boost productivity, and reduction of salinity. But sometimes these ecosystem services comes with a trade-off of reduction in water quality. In addition to that, the sea surface temperatures are rising in the area, and this change or increase in temperature might also influence the intensity of tropical storms as well. So because of the combination of all these climatic and anthropogenic changes, Louisiana estuaries are going in profound alterations. But what is unique about these changes now yielding is that they're creating an optimum conditions for cyanobacterial blooms to occur. Because we are freshening our estuaries, we are introducing a lot of nutrients, and cyanobacteria likes warmer temperatures. So when this whole package comes together, then we started seeing quite a bit of toxic cyanobacteria blooms in different parts in the Louisiana estuarine systems. So I'm sure you're familiar because you are experiencing that as well from time to time. But cyanobacteria is a photosynthetic bacteria like land plants or algae in the ocean, in the aquatic systems. And their bloom or increasing in biomass becoming really problematic because they are capable of producing multiple toxins when they're abundant. Not every species produces the same toxin. So there's a diversity of toxins the system can get exposed to when they are increasing. And also in general, compared to diatoms, they are not the best quality food to support higher trophic levels. And many estuaries in Louisiana, the Barataria Bay, Breton-Sound Estuary, and the Lake Pontchartrain Estuary are common places we see annual cyanobacteria blooms. And there's a lot of connections and discussions that river diversions is supporting these blooms. And it could be an easy answer if that was the case, and it's still very much linked and supports it. However, the connection of these river diversions to formation of cyanobacteria blooms are not as straightforward that we think. So I'm hoping that I can give some examples to be able to support that statement. But before then, I just want to tell you how we started working. So we started looking into Lake des Allemands, which is the very upper part of Barataria Bay, that doesn't have any river input directly. It collects a lot of wastewater treatment, treated waters, agricultural runoff. So it's a very hyper-eutrophic system. And we started looking at it because there was always a green color all over. And when we started looking at it, that was my first introduction to cyanobacteria. We have seen microcystis, which is one of the cyanobacteria species, present throughout the year. And then we had seen another toxic species, Anabena. Now people call Dodicospermum. So it used to be Anabena, it was much easier. They are sometimes coming up and then going, and then the toxin is there. And the toxin is not only in the water, but we have detected quite a bit of high toxin levels in blue crab. So we have seen that this is real, it's happening. We were hearing in other conferences that it was happening in different estuaries, but now we started seeing in Louisiana estuaries. And This is not something that we discovered in 2008 or so. There was more before then. But that was our first introduction, how intense they can get. So I was pretty impressed. And we started a little bit looking at the food web interactions. We know that the blue crab can accumulate quite a bit of toxin. And the catfish, we found several dead catfish, and we did detect toxins in that study as well. And their connection to benthic food web, the intermediate vectors that can directly feed on cyanobacteria. That's something that we started working, but this is something that we would like to expand in the near future. The food web aspects of cyanobacterial blooms is not quite studied in Louisiana estuary, so we are hoping to revisit that. But today I will focus on examples of two estuaries that we experience cyanobacterial blooms, but they are quite different from each other morphologically, how they connected to river, and the structurally how they experience cyanobacterial blooms. One of them is Breton Sound estuary, and this estuary receives river water from Carnarvon diversion, and it has a salinity gradient, so it's a very fresh upper estuary, goes down and meets with the coastal waters, and where these are the oyster beds are located in this area. It's a very vegetative wetland around it. And the river flow is so much smaller, and how much volume that comes into this estuary compared to Lake Pontchartrain. And the purpose of the river diversion was originally to reduce salinity to promote oyster production in that area. On the other hand, Lake Pontchartrain estuary, it's an open water system, and it's an oligohaline, low salinity throughout the estuary. It connects with the river through Bonnecaire Spillway, and the purpose of this river diversion was to protect the New Orleans residents from flooding. And it receives a huge volume of river water when it operates. So I'm going to start with the Lake Pontchartrain estuary, and it's going to be a little roller coaster. But the point is, every time we study this system, we have seen a different outcome in terms of cyanobacterial bloom formation or biological response. When the Bonnecaire Spillway doesn't operate, it's closed. The system still can take nutrients from the tributaries in the northern part of estuary. Lake Marupas brings some nutrients, and the structure, the river diversion structure itself, it leaks. So there's a continuous low level of nutrient entry in this system, even the diversion is not operating. And depending on the wet versus dry years, the discharge from the northern tributary can be pretty, strong. And there's a tidal influence. So during these non-operational years, we see these nice kind of a succession of productivity that dominates with the cyanobacterial bloom in the summer months. Because of the high residence time, warmer temperatures, and remaining nutrients in the system support cyanobacteria in the late summer, maybe sometimes early fall months. So my graduate student, Nicole, right here, she joined with me. And she is actually studying Lake Pontchartrain cyanobacterial bloom dynamics in non-operational years. And this research was funded from Louisiana Sea Grant, and Matt Hyatt here, John White, and myself were the PIs, and Nicole was the graduate student, and she's about to graduate. That's what I'm saying, was almost done. But she's looking at the biological kind of dynamics of the system, and Caitlin is looking at the water flow and residence time in the estuary. So I have just a few slides from their study to be able to present what kind of dynamics we see when the diversion is not operating. So Nicole separated stations. These are all of our stations we have sampled through 2021. And then we separated station based on their hydrological influence. So we had some spillway influence stations with yellow. We had tidal influences, orange, and then we have the tributary influence stations in red. And she looked at their environmental characteristics based on those influences, and then also look at the biological response, specifically focusing on cyanobacteria. So I'm just going to be showing you results from these spillway influence stations when the spillway is not open. So right here, I hope you can see it. This is the leakage, the discharge from the spillway when it's closed. So there was a leakage, there was a peak on the discharge in March and April in the springtime. The figure on the left is PC to chlorophyll. PC is a phycocyanin, which is an indicative pigment for cyanobacteria in freshwater estuary systems. And chlorophyll is the general pigment who kind of represents the complete biomass. So when the PC to chlorophyll ratio goes up, that indicates the domination of cyanobacteria in the system. So we have seen that cyanobacteria, like I mentioned just before, expectedly increase in their biomass in the late summer months. Almost all the stations except this one, which is the further away from the spillway. But what was interesting is this station, D2, which is the closest station to the spillway, we not only have seen bloom in the summertime, but we also have seen bloom later in the year, November and December, when we did not see that in any other stations. So the leakage really is influencing the dynamics of cyanobacteria in the system. But what was even more interesting is the figure on the right is the toxin microcystin. It's one of the common toxins they produce. And the levels are there, but low. Throughout all the stations that we looked in the spillway influence station. But here, D2, when we experienced 2 blooms instead of 1, the first bloom yielded lower concentration, but the second bloom in November and December corresponded to very high concentrations. So this is the first time, usually when funding agencies interested with Lake Pontchartrain when there's an event. And during the event times, we study it, but then we never had a chance to look at the baseline or when the spillway is not operating. So this is the first study Nicole is kind of opening up and showing that we do have these problems, even the spillway is not open because of these different resources of nutrients. And the toxicity can be actually quite high, which creates an environmental concern. So when the Bonicare spillway is open, which in the past didn't happen all the time, it opened maybe for every 4 to 5 years, depending on the Mississippi River water height. But if you look at the recent openings, it's almost every year, and in sometimes even twice in a year that we experience, and I'm sure you're all aware, in 2019. And when it opens, it creates a very strong plume, sediment plume, that separates the estuary basically into 2 sections. Inside the plume and outside of the plume creates a different dynamics. So this is a figure from Caitlin, the other graduate student in the project, that shows all the opening times. As you can see, there is a huge variability when they open and how long they stay open. So this is from 1945 to 2020. So there's a great variability. What she did is she picked the 2018, which is the purple one. It opened in the month of March. And she stimulated to look at the bloom formations. I hope it will. I thought it was so cool, so I wanted to share, and she was happy to share with you all. So this is where the salt water comes in. But when the spillway is open and the plume is forming and it becomes so overwhelming it pushes everything out. But I will show you at the end something is very interesting. So it goes all the way, but then this area in the northern part of estuary kind of gets disconnected. So that area, the water residence time is a little bit longer, which allows cyanobacteria to form. Even the rest of the estuary is too turbid for algae or for any phytoplankton to grow. So therefore, many times we always start seeing these blooms in the northern part of the estuary, and it kind of gets expanded based on the wind and the water residence time at the time of the sampling. Okay. So when the river structure is open, then we start seeing these things. As I mentioned again, there are differences in the timing and the duration of the opening, and the volume comes in. And in the past, in many times, the expected opening times usually corresponds to spring, when the water gets high. So I'm going to give you two scenarios. When the river diversion is open in the spring, what happens? One of them is 2008 opening. This corresponded in the springtime. It stayed open about a month. And what we have seen, there was a very clear plume formation that spreads the phytoplankton biomass from the rest of the estuary. When the diversion was closed, plume collapsed, and then the primary productivity kind of expanded throughout the estuary. When that happened, we start seeing succession of phytoplankton species. When the light increased, turbulence got done, we started seeing diatoms and chlorophytes are taking advantage. And then when the system become phosphorus limited, then we saw a shift from diatom to microcystis. Microcystis is a toxic cyanobacteria, and it's not nitrogen fixing cyanobacteria, so it still needs nitrogen in the system, and it has an ability to store phosphorus when it gets limited. So in those conditions, they were more successful than diatoms. And then now, at the end, we have a system that it's both nitrogen and phosphorus limited. So there's a time lag that another biological response hits in. And when we start having cold fronts and hurricanes, the resuspension of phosphorus from sediments back in the water column stimulates nitrogen-fixing cyanobacteria so that they are not relying on internal pool of nitrogen. So Dodicospermum and Cylindrospermopsis are the two species we see usually. So in this scenario, cyanobacteria bloom was in the summer, and the phytoplankton succession occurred throughout the time. And this is a figure from Roy et al. Showing that the availability of both nitrogen and phosphorus during the opening, and the sharp decline due to biological uptake, but the nitrogen doesn't come back to system, but phosphorus does through the benthic flux that stimulates these nitrogen-fixing cyanobacteria. The second scenario was a couple of years later in 2011. The expectation of 2011 was it was the largest nutrient flux. We expected huge harmful algal blooms, and we were having concerns about the fisheries shutdown. So this is a comparison between the 2008-2011. You know, it's in the spring, early summer. If you look at the volume of water, the 2011 had huge amount of volume with lots of more nutrients. But I will just cut this two part over here and then go to the next slide. If you look at the concentration of biomass in terms of phytoplankton, they were about the same. Even the second one had much more nutrients coming in. And look at the composition. When we have seen cyanobacteria in the summer following a succession of different species, we have seen no cyanobacteria blooms in 2011 ever. And this was interesting, and we didn't know what might be causing the absence of cyanobacteria, and we look at some of these tributary discharges compared to spillway. 2011 was the lowest discharge coming from northern tributaries compared to some of the previous openings, and that brought us a topic of thinking that might be the seed population actually coming from these tributaries, placing themselves in the estuary, and when the conditions are right, it blooms. And when the discharge is low, maybe there is not much of that population settles back in the estuary. And then 2016 happened. And this time, the first time ever, we had this very, very early spillway opening in January. It was a short period, pretty, short, but good amount of volume came in. And most likely due to cooler temperatures and low light availability, there was no cyanobacteria blooms. The overall biomass was also pretty low compared to other opening periods. So there was a low primary productivity, and then all the nitrogen that came in the system slowly left the estuary and kind of transported to coastal ocean. And actually there's a group of you published a paper in 2020 showing that the reason why this nitrogen displacements took longer than it's expected because wind was pushing water and keeping it stay in the estuary before it goes out in the coastal ocean. So it took quite a bit for that nitrogen being transported. So the wind plays an important role. But again, biologically, there wasn't many activity, excitement going on. However, this can change. If we start seeing warmer temperatures in winter months, then we might see these cyanobacteria dormant cells that is staying in the sediments when the temperatures are cold and there's not enough nutrient. They might just come back and regerminate much earlier than the times before. So because nutrients is already there, it's all about the temperature as a stimulant, then they might be able to regerminate. So I had an undergraduate researcher in my lab a couple of years back, and we did a study. We looked at this phycocyanin in the sediments. This is November and December of 2019, and this is February and March of 2020. And we had seen quite a high phycocyanin concentrations in the sediments. And then she took the sediment from March, brought back to the laboratory, and placed the sediment in a filtered estuarine waters with the same temperature. The control was no nutrient enrichment, and the treatment was the nutrient enrichment. And she saw that with the nutrient enrichment, the sedimented cyanobacteria came back to water column and increasing their biomass over time, and which we didn't see that in the control samples. So this is another area we would love to expand and look into more in terms of winter/spring dynamics. The last example in Lake Pontchartrain I want to show you is the spring and summer, which happened in the 2019. And it was always a super unique year. It opened for such a long time, and it lasted to the warmer months in the summer, which is critical for cyanobacteria to take advantage over other phytoplankton. During the opening, the volume that came in was about 28 or 29 cubic kilometer, and the estuary itself can hold 6 cubic kilometer. So all that water just flushed out, as you know and experience, and these basically nutrient-rich fresh water settle in these coastal waters of Lake Borgne and Mississippi Sound and cause an intense cyanobacteria blooms in that area. And we were collaborating with Rick Stump from NOAA, and he has an amazing webpage that anyone can go and look at these bloom dynamics in the Lake Pontchartrain estuary basin. And you can see there was a huge transport of cyanobacteria blooms outside in the coastal waters. As soon as the second opening closed, for the end of July, we start seeing the formation of cyanobacteria in the northern part of the estuary. And the changes in the wind strength, this bloom got expanded to the other parts of the estuary. What was super unique about this is we did not see no bloom. We did not see a succession of species. We have seen coexistence of both microcystis and dodecaspermum. This is extremely important because there's no nitrogen limitation, there's no phosphorus limitation, and I can actually go, there was a Hurricane Barry, if you guys remember, that kind of caused a peak in the phosphorus, lots of external nitrogen, temperatures are warmer, so both nitrogen fixing and non-fixing cyanobacteria were able to coexist in that environment. What is kind of concerning about that is Anabaena can produce more than 3 different kinds of toxins. And microcystin is producing microcystin, and dodecaspermum is able to produce microcystin, soxytoxin, anotoxin, and so forth. There's a couple others. And so when they are coexist, then they can create a huge environmental health risk, also for the human health risks as well. On the other hand, Breton Sound estuary, so just shifting gears a little bit because that's also an estuary in Louisiana, also experiences cyanobacterial blooms, but it's more seasonal because the river diversion, it's not as variable that we have seen in Lake Pontchartrain. This is an annually and seasonally operated system. So we see these diatoms and chlorophytes in the springtime and cyanobacteria in the summertime that corresponds to high temperature, low discharge times. And when they are high in abundance in the late summer, fall, then we also see corresponding toxin production. But what is interesting in this system is we see higher presence and toxicity when you get away from this river diversion. And what it means is during periodic high flows, the cyanobacteria that accumulates in the upper estuary can be transported to lower estuarine areas. And we know that cyanobacteria can adapt to low nutrient conditions as well as moderate salinities. So if that happens and they're transported to lower estuary, then they can create a potential contamination problem for oysters. This is an experiment, it's not published, but we kind of trying to do some experiments with oysters in the lab many years back. But we have seen the oysters were having no problem grazing on or taking up dodecaspermum in a very high speed. So that's another food web related aspects that we want to focus. I think we understand the complexity now, how these systems are influenced from these river diversions and how cyanobacteria responds. I think the next step for us to kind of look into environmental impact and human exposure risks that comes from the food web contamination. I don't know how we are doing in time, so I was just expanding some, but I know we started late, so if people are starving, I totally understand. This would be a good time to stop if it's needed. You tell me.
Do we have any questions? Or do people want to keep going?
No, I do have a question on that.
Sure.
On the spillway. So on the spillway side, when the spillway didn't work or stopped, just not operating, you still see the cyanobacteria increase, right? But what's interesting is that the late summer, actually late autumn, you see a second peak. Right. But if you look at the leak of the water, it's pretty much in the early March, right?
I know, exactly. So we didn't, obviously the leakage was much earlier that fueled potentially the August bloom in the summer. But what fueled the November, December, we don't know. Maybe.
I think it's really like this comes with the residence. So like when the spillway is not open, you have a lot higher residence time going on. So anything that's coming.
Nicole, what was the species? Do you remember the one in November and December?
It's mostly animated.
Yeah, if it's mostly dodicospermum, that we call it, I keep mixing too. But those are nitrogen fixing. So if there was some rich suspension of phosphorus in the area, then it might fuel the dodicospermum bloom, even the nitrogen is not there. So the external nitrate comes mainly from that leakage. And in the absence of it or lack of that, might promote still some of those cyanobacteria species like dodicospermum. On the other hand, the other species of cyano, as well as the diatoms and chlorophytes, might not have a chance to bloom.
That experiment only 1 year, or is every year you found it? Just 1 year?
Yeah, that's what I'm saying. We always got funded when there's an opening. Because everybody's so interested in what's going to happen. So we need more funding to be able to understand the non-opening bloom dynamics and what fuels them. And this was our first kind of a full-time opportunity to sample the entire estuary and look at some of the residence times and the nutrient dynamics. So I'm excited to be able, if we can expand that.
Okay. You know, the other question, just physics. What's the typical residence time of water in the deep function?
I think it's a couple months. Yeah, I think 3 months.
3 months.
Yeah.
That's without an opening, but once it's opened, it goes down to like a week or something.
Comes and goes.
Days, yeah.
Yeah.
One question on the potential impact on the oysters. The Mississippi River has a very high alkalinity, and we actually looked in the Mississippi Sound when the 2019 opening was happening to see if, potentially it is a buffer to ocean acidification, but it seems like in the Mississippi Sound, at least, the drop in salinity is more harmful to the oysters than anything else happening.
Sure.
So yeah, what is the biggest risk for the Louisiana oysters when there's an event like this?
I think cyanobacteria is a bonus. Salinity is a huge issue, like you indicated. But when the salinity is down, there's also always a potential for cyanobacteria to bloom. In terms of the toxin impact on oysters, I don't know if there's a direct, like a negative impact on the oyster. But the oyster filtration rates are super high, so they can filter everything around them in a very short period of time, and there's a direct human contamination if that happens. So it's more kind of risky for people that consumes them. And these type of toxins, any algal toxins, they're pretty resistant to heat. So cooking the oyster might not get rid of the toxin. Like sometimes you do that for bacteria,
Right.
but not for algal toxins. And one other danger for oyster, which was a couple slides that I have added over there, they are in a location that they can get exposed to more freshwater toxin as well as the marine toxins. And toxic diatom pseudonetia, which I worked many years, it's also present in Louisiana coastal waters. And they're also tolerant to certain level of drop in salinity. So then you end up in an environment with oysters that have both freshwater and marine toxins, and you don't know which one is being more harmful or get accumulated, but that also creates a higher risk for human exposure as well. So our systems are very much freshwater to marine continuum. I mean, everything just has exchanged, and it kind of levels up the risk factor because of that.
Sure.
You mentioned the dormant cysts that'll sit on the surface of the sediment. Do you have any more information on those? Do you know anything about the size spectrum they're in?
This was like a opportunistic undergraduate work. If Nicole comes back for a PhD, that's we are hoping we're gonna work on. But what we wanted to first see if cyanobacteria is there. And the easiest way for that student was to get a sediment sample and look at the phycocyanin pigment. And the levels were pretty high. So they are definitely settling down. Whether they are settling down in a perfect dormant cell form, I don't know. Because usually we do get bloom in late fall. This could have been a dying bloom settling down, or it could have been the dormant cells that they actively formed. So that part we didn't do it. But she also detected toxins in those sediments that I didn't show. So that's another level. And I don't know so much the difference between the toxin production happens in vegetative cells versus if that actively happens in the dormant cells. Probably it's different. So that a lot of things to tease out, but I think the critical thing is they are there, settling down. These are shallow systems, so easy to back in the water column when the conditions are right. And we start seeing higher temperatures in November, December. So it's just gonna happen. I think the matter of time that we start seeing these blooms now in the winter time even.
All right, we could take questions from online if anybody has one, or anything else in the room.
No, I just, how do you feel? Is it safe still eat the oyster?
Oh God, you're asking the wrong person because I don't eat anything that filter feeders. So any organism that filters things, I don't. But if you're careful and choose the right timing in the year, I'm sure it's safe to eat.
What's the right time of the year?
So I'm just telling that cyanobacteria loves the heat. So the summertime is not a good time for the consumption of these organisms if they are exposed to these blooms. But I have to be very careful because I know everybody loves seafood. I'm practically vegetarian, so I don't eat much meat. And the filter feeders, it's not just one factor. I mean, they just filter without too much selectivity. So there's heavy metals, there's pathogens, there's toxins from algae. It's all that lovely stuff. But it's important also how quickly they can depurate. So they can take in, and if you're unlucky, you eat, you get sick, but they can depurate quickly too in many cases. Then if you're lucky, you can eat the time that they depurated, then you're fine,
You don't eat the filtered fish because your profession, right? You see too much horror stuff.
I just know little, yeah, exactly. But I try not to tell it to my kids and husband so much, not to influence them. But yeah, no, we don't.
All right, if nothing else, then we'll thank our speaker again. I'm so sorry for the delay, but that was a great seminar. Thank you.
Thank you, appreciate it.